introduction, and I'm excited to talk to you all today and tell you about kind of my perspective on cell therapy and my experiences in working on cell therapy for oncology. So today I know we have kind of a broad background, broad audience, so I want to be sure to keep everyone on the same page and give some solid introductions to cancer, to different therapy modalities, to really introduce immunotherapy before I get into where we're at with cell therapy, some of the major challenges for the field, and then to kind of end by giving some of the scientific background on the cell therapy projects that are ongoing for cancer at the GBF. So I think when we think about cancer, we definitely all think about kind of this figure on the left and sort of the causes and things that we try to avoid getting cancer. But I just wanted to be sure that we cover the definition of cancer. So in order for a cell to become malignant or to qualify as cancer and to create a tumor, these are all words that we know, it really has to arise from a genetic mutation that can be caused by many of these external sources and even just the process of cell division. So built into every cell as it divides, there's plasticity, which allows us to evolve as organisms, but also inherent in that process is the possibility of a genetic mutation that would allow a cell to either infinitely divide or not to die. And so that cells can accumulate in tissues and cause cancer, cause a tumor, basically make normal cellular function not doable. And this can be fatal, as I think we all have experience with. So generally, when someone is diagnosed with cancer, there are many different therapy options, and they're here depicted in all of these different colors. And I think the main point that I wanted to make here is that for the most part, from hormone therapy to surgery through to radiation therapy, most of these treatments have really been aimed at targeting those malignant cancer cells, right? And trying to eradicate those cells, to get them to stop dividing, to get them to die through these various modalities. But one branch that in some cases has been, the idea to use immunotherapy has been around for decades, but the practical actual application has really taken off in an amazing pace that I'm gonna talk everyone through. But I think I just wanted to point out that when we think of a tumor and the way that a lot of drug development has happened, it's been really focused on thinking about these malignant cells. So the cells in this very simple diagram that are depicted in as these kind of gray and white, strange looking cells. However, when you actually look at a tumor, there really is this whole what's known as the tumor microenvironment. And within a tumor, you have things like these support cells, these fibroblasts, that within a tumor really are quite dysplastic. So they're also quite different from what you would find in normal healthy tissue. You also find, depending on the tumor type and depending on the stage and the grade and everything of the tumor, you can find various immune subsets that are here depicted in these different colors. And these cells can be at varying degrees of sort of normal functionality. But I really wanted to make the point here that the tumor microenvironment consists of many more cell types than just the tumor cells themselves, and that immunotherapy, the whole point of it is to harness some of these other cell types and to use them to actually combat the tumor and slow up the tumor growth process. So here's kind of a timeline of cancer therapy treatment and sort of the development of various modalities. Again, just to highlight that most of these therapies, everything from surgery through to targeted therapies that has been a really exciting field since the 80s, have been really focused on the tumor cells themselves, right? And trying to get those cells to die and to go away. And immunotherapy is really this new field where we're going to be harnessing the immune system. I also wanted to make the point that for the most part, for a fair number of patients who are diagnosed with cancer, the mainstay of therapy still really would be surgery, chemo, and radiation. These kinds of therapies that have been around now for, centuries. And when we think about all the time and effort and work and dollars that have been put into developing cancer therapy and understanding the cancer genome and everything about cancer biology, we're still clinically really using a lot of therapies that have been around for centuries. And basically using some of these very old therapies and some of the newer therapies, I just wanted to highlight that even in 2020, when we know that COVID-19 has really been dominating the news, and we really think about that, I don't know, this figure to me was, even as a cancer biology researcher, this figure to me was sort of, I don't know, startling or like a real reminder that heart disease and cancer still really are the top causes of death in the US. COVID certainly is shocking and awful in its own way and came from 0 to this, astronomical number. But I just wanted to highlight that even with these different therapies, these different modalities, cancer is still the number 2 cause of death in the US. So there still is a long way to go in terms of getting these rates down and finding cures for cancer. So when we think about these therapies and we think about all of the work that goes into understanding cancer and developing new drugs, I had fun sort of looking into this and thinking about how are we really doing in terms of tackling cancer mortality as an issue in our country. And you can see that this is looking at cancer mortality rate on the y-axis per 100,000 people and over time. And you can see that this is obviously taking population growth out of the equation. You can see that the mortality rate has been decreasing over time, certainly in the last couple of decades, which I think is encouraging. I think it's also important to note that the major decrease in mortality rate is really related to a decrease in smoking. And you can see this by the top green line represents lung cancer in men over time. And decreases down here, you can see the next most common types of cancer are breast cancer and colon cancer as sort of these high mortality cancers. And you can see that they have somewhat of a downward trend over this time frame. And most of that has been attributed by epidemiologists to more screening rather than therapy. So this isn't meant to be depressing in terms of how we're doing on developing novel therapies, but it's just to point to the fact that we still have a long way to go and there's still huge unmet need for cancer. And maybe this is looking at it from slightly different angles. That said, I think there has been a whole blossoming of this field of immunotherapy over the last decade or so, and that's really been highlighted, and there have been a lot of advancements, and they've been highlighted not only in scientific journals but also in the common press. And I think more and more, as we see various family members or friends who are diagnosed with cancer, certainly immunotherapy is becoming more of like a household term. So I do want to dive into immunotherapy and talk a bit about it, and then, give you an intro to cell therapy from this angle. So in order to do that, this is known as the tumor immunity cycle. Basically, you can't go to an international conference in cancer biology or immunology or immunotherapy without someone showing this diagram. Often you'll see it in many talks. So it was developed by Dan Chen and Ira Melman. They're two big players in the field. But essentially, this is the path that's necessary for the immune system to handle a tumor. So basically what happens, as we covered on the very first slide, tumors have mutations in their genome. And as such, then they have mutant proteins. And the mutant proteins can get expressed on the surface of the cell. They can be shed, as is depicted here. And as they express these mutant proteins, the immune system, so these are called antigen presenting cells, and they're part of an arm of the immune system that's known as the innate immune system, will pick up some of these mutant proteins, carry them to the lymph node, where then they interact with the adaptive immune system, so subsets of cells called T cells. And there the T cells are basically educated within the lymph node. And they're basically taught, this is a mutant protein and you need to act like a soldier and go out into the body and find the cells that contain these mutant proteins and kill them. And so then the T cells navigate through the blood vessel and have to infiltrate through normal tissue as well as into tumors. Then through molecules expressed on the T cell surface and on the tumor surface, they're able to recognize those mutant proteins that they've been trained up to recognize, and then they can kill the cancer cells. So I think it's just a phenomenal, amazingly elegant system that we've all evolved to have. But these are the 7 necessary steps basically in order to control a tumor. And the reality is that we all have mutant proteins that arise during cell cycle, and in a healthy, normal state, our immune systems are able to control those cells that have acquired mutations, and our immune systems are able to prevent the growth of tumors. So when we think about immunotherapy, there are actually several different modalities that have been developed over the last few years. And in addition to the tumor immunity cycle coming up in all international conferences, you also have lots of vehicle analogies. It's like the field loves these vehicle analogies, so I'll use that. So if you think of the immune system as like a race car, and you want to keep your race car healthy and in check, then these modalities can come in. So there's a whole field of cytokine therapies that have been given, and I have one slide about that where we can talk about that a little bit, but it's essentially like giving the car more gas so that it can go fast. Then there's a whole field of immunotherapy that are called checkpoint inhibitors, and there basically you are inhibiting the brakes of the race car so that you're allowing the immune system to go and getting rid of the brakes. There's also therapeutic vaccines, cancer vaccines, that I'm gonna draw the analogy and call it like taking your car to a mechanic, so being re-educated and taught to perform. And then adoptive cell therapy, which is essentially just getting a whole new race car. And that'll be, the main crux of this talk. So here's another timeline. I just love medical history, but I won't belabor it here, except to say that basically all of these different modalities have been in development. They've been postulated and were proposed, a long time ago, as early as the 1800s, when Dr. Coley gave he inactivated bacteria and actually was able to ramp up the immune system of some of his patients and saw really good regressions in some particular subsets of sarcomas. But then you can see into the 80s and 90s, there were some cytokine therapies, and I'll show you some of the plots for those and what really spurred people on to think that immunotherapy really truly can work clinically. The checkpoint inhibitors came in right in the mid 2000s. That's when I was working at Regeneron, as Matt mentioned. And so we were doing a bunch of combinations with some of these checkpoint inhibitors and targeted therapy, so that we were both targeting the tumor cells as well as trying to release the brakes on the immune system. And then the cell therapy field, I will get into as well. And it sort of looks here like the different modalities are staggered time-wise, but the truth is there's cancer vaccine trials that are going on now. So they're all still, all of these fields are still very much alive and well and in active development. Right. So I think I wanted to just give you a couple of examples of some of these other immunotherapies, and I think they help set the stage for why cell therapy was hypothesized to work and why it has been successful. So this is one example where this is the idea of giving the immune system more gas. So IL-2 is a cytokine. It's a particular type of protein that's found within the blood, and it can activate many different cell populations. So it can activate those T cells that you see that I introduced in lymph nodes. It can also activate dendritic cells and NK cells. So many different immune subsets. And it was administered systemically to patients. And this is a plot. Okay, so these are Kaplan-Meier plots. Just for anyone who hasn't seen them, it's the probability of survivals on the y-axis, and time here in months is along the x-axis. And so at the beginning of the study, 100% of the patients are alive. And in metastatic melanoma without treatment, this line would go all the way down to 0 by 60 months. So that's sort of where things were at for metastatic melanoma. And this was, again, it's just giving IL-2 that's ramping up the immune system. And this was where clinicians saw that there was this survival tail. So there was some percentage of patients that were able to respond, where activating their T cells and activating their immune system allowed them to reject the tumor enough so that they survived. And this was also found, it's kind of a similar sort of plot. This was renal cell cancer. And so you had 200 patients who were not responding, but 21 patients who had a complete response. And so this points to the fact that, I mean, I think scientists wanted to figure out who are these patients, right? Why are they responding to IL-2? What is it about their immune system? What is it about their tumor cells? What is it about their whole immune system that allows them to respond so well? And if that could be understood, then you would know what patients to put on an IL-2 kind of therapy. And also maybe it could help you figure out other ways of like inhibiting some other pathway that might be preventing this kind of response. But I think this was really an exciting clinical finding, even though it's a small percentage of patients, just that this approach, this activating the immune system, could work and could be curative in some of these indications. And then this carved the way for the idea of checkpoint inhibitors. So again, this is where you're inhibiting the brakes of the immune system. So here you can see in this picture on the left, as a tumor cell, it can be an antigen presenting cell or a tumor cell, but we'll keep it simple with just thinking about it. As a tumor cell interacts with a T cell, it sees the red is that says AG, that's antigen. So that is that mutant peptide that I was describing, or the mutant gene that's expressed in the tumor cell. And as the T cell sees it, it gets activated by these blue and purple signals. It needs both of these signals. In Immunology 101 classes, it's called signal 1, and then signal 2 is this activation step. However, there's also these inhibitory proteins that can be expressed by tumor cells, and they can upregulate. Here it's in red, it's called PD-L1, but I'll show you on another slide. There's hundreds of these molecules that can be upregulated to silence the immune system. And so here the tumor cell expresses its mutant protein. The T cell wants to kill it, but then the tumor cell also puts on the brakes on the immune system and tells the T cell, don't kill me. And there have been many different drugs that have been developed now. These are 2 antibodies that have been really developed in this field to block the brakes, so to inhibit the brakes. And these have been really very exciting, again, in melanoma. So here's that historic red line curve where without therapy or with historical treatments, basically within 5 years of diagnosis, things weren't good for patients. However, with CTLA-4 is one of these brakes molecules. If you put in an antibody against that, you got, again, one of these kinds of tails where some patients were responding. And then PD-1 is a different molecule. And then here, when this figures from 2016, these combination experiments have been done clinically, and the dashed line pretty well represents how the trials actually turned out, where you could combine these and shut down both of the breaks and get really a very significant response in, close to 50% of patients, which, as this was presented, everyone's, jaws were dropping, and this was really exciting, meaningful responses in the clinic for an otherwise incurable disease. And there's still, probably thousands, I would say, of clinical trials going on for checkpoint inhibitors, and trying to figure out where they can work and in what indications they can work. I will say, I think melanoma was an exceptional place to find it for a variety of biological reasons. There's some subtypes of colon cancer that also respond very well to these checkpoint inhibitors, some lung cancer. Anyway, all to say, there's more work going in figuring out what types of indications can respond and what can't. It's not a magic bullet that cures all cancer, and doctors and universities and pharma companies are learning where they're going to have most success in this field. Oh yes, and then this slide is just to overwhelm you and show you that it makes sense that they're not always going to work, because there really are so many of these checkpoint inhibitor molecules. The system, the way all biological systems, it's evolved to have lots of redundancy. And so you can block PD-1, you can block CTLA-4, and you're still going to have other compensatory mechanisms that can allow a tumor to block and shut down the immune system. Drug companies are developing these kind of mixtures of many different antibodies. But at the end of the day, it becomes a little bit like whack-a-mole, and it's a complex puzzle. Okay, so with I think the excitement about the clinical findings with cytokine therapy, with checkpoint inhibitors, I think it's clear that the immune system is super powerful and can be harnessed to do amazing things clinically in fighting tumors within patients. And that all is sort of maybe a helpful introduction to what cell therapy can do. So there's really two types of cell therapy, and we actually have projects in both, and sort of one project that's both fields overlapping, which is exciting. But basically you have non-engineered approaches where you're essentially expanding you can expand different types of cells from within a patient, and then in a way expand them, sort of clean them up, help to supercharge them, and then put them back into a patient. And then there are engineered approaches, and this is where you get into some of the CAR T cell field and CAR NK cells, for which we have two projects at GBF. So I'll get into that. And there you are using, you're actually engineering the cells and putting in a genetic modification so that they will be then specific to the tumor. And I'll talk about that a little bit. So for the non-engineered approach, this is a simple depiction of a process for something called TIL therapy. So those are tumor infiltrating lymphocytes. And the idea is that within a tumor, you do have T cells that would be reactive against those mutant proteins in your tumor. But they're just, for a variety of reasons, and it's some of those checkpoint molecules and a variety of other mechanisms, they're not able to function, or maybe there aren't enough of them, like the tumor has outgrown the population of T cells. And there is a field that was really pioneered by a guy named Steve Rosenberg, who's at the NIH, where you can take out a tumor, you can isolate and grow up the T cells that are there, and then you can reinfuse them. And I'll show you the approach that's taken by Eduardo Davila's lab and Liz Franks, who's also new to GBF. And I'll tell you a little bit about their project on a later slide. But this would be considered a non-engineered approach because you're just expanding the cells. You're not actually changing anything within their genome. Then to introduce engineering approaches. So CAR T cells are just one example of an engineered approach, but they are the most common engineered approach. So I'll talk you through this. Basically, again, we would isolate here, instead of isolating the cells from the tumor, we isolate them from the blood. And so we don't expect that these T cells are going to be tumor reactive. They're just your T cells. And in the context of everything that's going on right now, they're maybe mostly specific to COVID-19, either from vaccine or from exposure. Anyway, these T cells can be specific to any virus, bacteria. They could be naive and never having seen anything, but we take T cells from the blood. And then we introduce a gene that's going to be converted to a protein and expressed on the surface that will allow the T cells to now recognize and kill the tumor in the presence of a specific protein expressed on the surface of the tumor cell. So we engineer them, we grow them up, and then we reinfuse them. And just to describe the kind of engineering, this is the basic design that's used for most of these chimeric antigen receptor constructs. So you have what's called the single chain FB on the surface. And this is what gives the T cell specificity and allows it to bind to the tumor cell. And then that is fused through what's known as the hinge domain to the intracellular portion of the T cell. And there you have, again, the signal 1 and signal 2 that I introduced on the T cell slide. And so this is really synthetic biology at its finest. It's taking proteins that are expressed in normal cells, understanding what domains are needed in order for the specific cell signaling and the processes and the functionality that you want to get out of the T cell. So signal 1 is, and most commonly, I think in every, certainly in every FDA approved CAR, but it's derived from the CD3 zeta molecule. And this tells the T cell to release its cytotoxic granules and to actually kill the target that it's in contact with. And then signal 2 is a co-stimulatory domain. And this is derived from a bunch of companies typically either use a molecule called CD28 or 41BB. Actually, I authored a patent. We looked at 20 different co-stimulatory molecules and found that they worked in various ways and to various degrees, depending on what you were looking at, and whether you looked at the T cell's ability to secrete cytokines or the T cell's ability to survive after it kills. But basically, this sends a kind of survival and persistent signal into the T cells so that it doesn't just kill the target, but it can divide, make more of itself, and persist for long enough to kill the entire tumor and to even kill a tumor as it starts to relapse or regrow. So there's been significant clinical progress, particularly in the CAR T field. And I think most people saw probably in the news last week, it was really the 10-year anniversary of remission for leukemia patients who had received some of the original CD19 CARs. So CD19 is a molecule that's expressed on leukemias and lymphomas. So that scFv portion targets the CD19 on the cancer. And there have actually been several approvals over the last few years, which has been really exciting. And these have all been, all except for this last one, have been around CD19 CARs. And then I thought this was a particularly interesting chart that I came across, that even in the context of COVID between 2019 and 2021, you can see this really rapid growth in active cell therapy trials. So it's a really exciting time where there's a lot of development in cell therapy, and you can see the number of CAR T trials has really doubled. But then you've also got these other more kind of interesting next generation sorts of therapies. And I'll get into that a little bit and kind of the benefits of thinking beyond CAR T. So I think now I just want to get into some of the major current challenges in the space and sort of approaches that companies and academic groups are getting at to really move the needle. I think the CD19 experience has been really exciting. I'll show you some of the progress that's making way to develop kind of the next generation tools in the context of CD19 cancers. But I think, you know, a huge burden, a huge unmet need in the cancer field is in solid tumors, in things like breast cancer, lung cancer, colon cancer, prostate cancer. And there, universally, CAR T cells have not been efficacious. And we can talk through a little bit what some of those reasons are and kind of the approaches that people are taking to try to scratch the surface in those fields with cell therapy. So the three major challenges, the three major buckets are manufacturing, efficacy, and safety. And I'll talk about those three over the next couple of slides. So when you think about manufacturing, when you think about CAR T cells, and when you think about manufacturing, and I think when you read a lot of the press in kind of the New York Times, the newspapers and things, is a lot of it's around how expensive cell therapy is. And can you believe that this got approved and it costs this much? But I tend to argue back, and I think, you know, we need to make everything accessible and keep costs down as much as possible. But this truly is a really expensive, cumbersome process. So as we talked about, the kind of process of editing the cells, you have to do a selection step, you have to expand the cells, and you have to reinfuse them. And the processes, as all of our operators at GBF can tell you, has to be done in very sterile conditions. Obviously, you don't want to be introducing any bacteria, any viruses, anything into the product, because it's going to be reinfused into a person. And this whole process typically takes, at GBF, our process takes between like 8 and 10 days. So growing cells and keeping them sterile for that amount of time is a huge amount of work and requires expensive machines and very well-trained operators. Another huge contributor to the expense is that it has to be, for autologous CAR T cell therapy, it has to be individualized. So the therapies are manufactured on a patient-by-patient basis. And then because the process is so expensive and individualized, it's obviously not available everywhere, and it's only available at specialized places like CU. But in order for it to be accessible to the whole, you know, everyone who has cancer, it needs to be, the process needs to be significantly improved. So some solutions that are being developed, there's an idea of creating off-the-shelf product. So it'd be great if you could just grab a vial and have CAR Ts that could work. So people are coming up with engineering approaches that would allow the T cell to be used not within the individual patient, but in anyone. And so you have to edit certain aspects of the T cell so that they don't attack the person who you're putting them into. And then there's a possibility of using alternative cell types, and I'll get into that a little bit with our collaboration with NKarta. Then another huge expense is the virus. So we use virus in order to introduce that molecule, the genes, into the T cell. And so there are several non-viral approaches. These are three that I'm familiar with. So there's electroporation, where you're introducing genes and shocking them into the actual nucleus. There's cell squeezing, and then there's nanoparticle approaches where you could, I think the nanoparticle approaches are really interesting because that's where you start to think about a concept like inpatient editing, where you could actually coat a nanoparticle with a protein that it would allow it to home to the T cells within a patient, get into it and actually edit the cells, but that's a long way off. So we still have job security at a cell manufacturing facility. I think the electroporation technique is the furthest along, and I know of several startup companies that are going to be testing their products. The capacity, the ability to engineer cells, it's much harder to do it that way versus virus, and you need enough of those molecules on the T cell surface in order to get good efficacy. Toxicity and safety. So this is obviously a huge concern. You can think about, as you engineer the cells, you can insert the scFv and those activating domains. They can theoretically be inserted into a gene that is necessary for the T cell to function properly, so that you could actually be causing a T cell leukemia in the product that you're creating. It's not common. It hasn't been seen a lot. There was one kind of famous report of it that was published by Carl June, where it jumped into what's called the TET2 locus. So it's happened. It's unlikely. But I mean, it's something that the FDA certainly wants to see evidence that it's not going on. Then you have these, what's known as cytokine release syndrome and some neurotoxicity that's caused by the efficacy of the T cells. So they're potent, they're going in, they're killing the tumor, and they're releasing all kinds of cytokines that activate the immune system. And it can be a huge stimulus for the system that, you know, was really caused actually fatal toxicities in some of the early days of CAR T cell therapy. Now clinicians know more about what they're looking for and how to treat it, and there are therapies. So in the context of CD19, it's not as fatal. It's not as fatal as it was at the beginning, but it's still certainly a consideration. As we start to think about new targets, you can have your scFv in the context of B cell leukemias. It's okay if you kill your normal B cells that express CD19. Any clinicians on the call, I guess okay maybe is maybe not the right word, because having no B cells in your system presents its own challenges. But if you think about solid tumors and you think about going after tissues like where you're trying to cure brain cancer, if your scFv is also killing normal brain tissue, that's obviously going to cause major toxicity. So this is a major hurdle in the context of non-CD19 malignancies. So there are several solutions that are in development. You can think about expressing something called a suicide gene, where if you would engineer that also into your engineered cells, so they would express the CAR. And then they could also express an inducible kill switch. And basically you could give what's depicted here as a dimerizing agent, but you could give some kind of therapy. You would give it systemically, and then that could activate a death pathway in the T cell. So that if you started to see a safety risk, you started to see toxicity, you could give this dimerizing agent and activate the killing. There's also off switch that would work somehow similarly with a dimerizing agent. And then these two platforms we were developing at Cell Design Labs. And actually that was the platform technology that we used to sell the company to Gilead. This is a technology developed by Wendell Lim. His papers are really cool. And this figure didn't come from them, and he's a total prima donna about figures, so he would probably not like this one because it's, I don't know, simple. His are very fancy. But basically here, the engineering is really awesome, where when CAR1 recognizes its target, there's a mechanical pull that's based on notch signaling, and that allows the transcription factor to go into the nucleus and to induce transcription of a second CAR. And this allows for safety, so that your tumor cell would have to express both antigen green and yellow in order for killing. And this would allow it to discriminate between normal tissue and a cancer cell. So these These safety solutions are all, I would say, really in development, although I think over the next year to 2 years, you'll start to see clinical trials with them, and you'll see how there's only so much you can learn in animal models, and there's some amount that needs to be discovered in the clinic. And yeah, it'll be really exciting to see how these platforms progress. So now just to get into efficacy, and I think this is an important picture to paint so that we can understand what's going on with the CD19 and CD19x22 trial that is happening at CU and that we're manufacturing cells for at the GBF. So simple picture, we can think of this CAR as an anti-CD19, but it can really be any CAR against a single target. And then thinking through the possible clinical outcomes. Let's say this is how the patient presents, and you give them the CAR T cell. And then you come back after some amount of time and you look, it's like here you're looking in the blood. And here you see a CAR T cell, but you see no tumor. So what does that mean? It means you've got a clinical response. Your CAR T cell got rid of your tumor, and over a long enough amount of time, you can call that a cure. Another possible clinical outcome is that you would have only tumor and you wouldn't have any CAR. And there you would say that your CAR T didn't persist, that it didn't hang around. A third option is that you have your CAR T cells are there and your tumor is also still there. But then when you look at your tumor and you look at the surface of it, you can see it no longer has that target 1 on it. So there's nothing for the CAR T cell to grab onto and kill the tumor. So the tumor is able to stay and continue to grow. And this can either be that the patient never responded, or this is what it looks like at the point of relapse. And it can be thought of as that target 1 is somehow lost over time, or, more likely it's that there was a small subset of cells at the beginning at the clinical presentation that didn't express target 1. And so those cells were never killed by the CAR T, and they are able to survive, and they're what induces the relapse. And it's in this context, this clinical outcome number 3, that the CD19x CD22 concept came to play out, and I'll show you that on a future slide. So here in 4, this is a head scratcher. We actually saw this with one of the solid tumor programs that we were collaborating with NIH in a solid where you had CAR T was around, but and the tumor was around, and the tumor still expressed target 1, but the tumor was still around, like you weren't getting any clinical response. And so this either suggests that the T cell isn't working, that it's not killing the tumor cell, and there's been some preclinical evidence of that the T cells can just be, either the T cells are sort of dysfunctional because they came from a cancer patient and had dysfunctional signaling, or that the tumor upregulates certain pathways so that it becomes like immune to T cell killing. They can be these interferon gamma responsive pathways that can get modulated in the tumor, and then they're no longer responsive to T cell killing. And then I think what we were seeing in this one particular clinical trial was that the CAR T cells were not actually able to get into the tumor and to access it. So again, that's getting at that whole tumor microenvironment, and can T cells even traffic there and get in? So I think that's what I wanted to highlight here, that really in order for a CAR T cell to work, you need a bunch to happen. It needs to traffic, it needs to recognize and kill properly. All of those pathways need to still be intact. It needs to proliferate and persist, and then it needs to counteract the tumor microenvironment. So there's all kinds of suppressive molecules in addition to those checkpoint inhibitors. There's lots of things that are shutting down the immune system. And I think I've covered most of this. So I'll just move forward, but basically there can be potency issues and then durability issues. So now just to jump in, the last section of this talk is just to jump into the projects at GBF. And I think some of these have been covered. They can each certainly be covered in their own whole talk. Today is just to give you an overview of the field. This is, yeah, and I think that Terry Fry gave a whole talk on the 19x22. Someone told me just before I arrived. So I just wanted to highlight the difference between the CD19 construct that's used in our clinical trial versus the ones at Kite and Kymriah. So they all have the same scFv, that surface portion. And then here at CE, we have the 4,1-BB co-stimulatory molecule, which is really thought to be helpful for long-term persistence. And then the manufacturing cytokines. So we talked about how much T cells like IL-2, but IL-7 and IL-15 have really been demonstrated to stimulate some STAT signaling pathways that are essential for memory preservation. And I was super excited when I was interviewing to find out that you guys are manufacturing, that we're manufacturing with 7 and 15, because I think it really preserves an important pathway. So we talked about how that target 1 can be cells that don't express target 1 can be expanded, and that would cause relapse. And that definitely happens in the context of CD19 therapy. So cells can, the relapse, and it's leukemia, but it no longer expresses that target, that CD19. And so another target that's on these leukemias and lymphomas is CD22. And so the idea here is that tumors will express, I've got CD19 in purple and CD22 in green. And so if you make a CAR that targets both, then you would be able to make a construct that has CARs for both CD19 and CD22. You would be able to target a tumor even when it's lost both either CD19 or CD22. And this trial we're just launching now, but it'll be really interesting to see, especially in the long run, how this works, how this dual CAR approach works, and are you able to circumvent that CD19 negative relapse that has been reported in the clinic. So then another project that we have at GBF is looking at a non-T cell platform. So it's engineering NK cells. And here the NK cells are engineered with a molecule on the surface. Instead of having this be a tumor homing and activating, engaging molecule, it's a molecule called NKG2D, which actually activates NK cells themselves and can turn them on, so that you could be activating other engineered cells and also other non-engineered cells that have a receptor for this activating molecule in the patient. And then they chose the OX40 co-stimulatory domain, and then they're activating their kill domain is the CD3 zeta. And they also have this membrane-bound IL-15, which is a cytokine that NK cells need in order for survival and proliferation. So they've got, this really nice mixture of molecules that we're engineering in. The process is somewhat similar to CAR T cells, although I think all of the operators at GBF would attest that this process in some ways is more cumbersome, more complex. It's not a closed system. And so it's been, and I was super impressed to learn the kind of expansion and proliferation experience that GBF is having. NK cells are notoriously challenging to grow up. And I think it's just really admirable and awesome that the process is up and really going as well as it is. So this is just to highlight that there are differences between T cells and NK cells. I would say one of the main, it's definitely more complicated to expand NK cells versus T cells. It's definitely harder to transduce them. All of those hurdles have just been phenomenal that GBF has overcome them. And then a huge advantage is that NK cells can be kind of one of those off-the-shelf products. So they don't have that specificity that T cells do, where you can give them in what's called an allo setting. So the product that's made at GBF right now is in clinical trial, and it'll be really exciting to see how those trials with this construct pan out. And then the final project, the final cell therapy for oncology project, is really run and spearheaded here by Liz Franks in collaboration with the Davila lab. So this is really exciting. It's taking a non-engineered approach of TIL and combining it with engineering. So the idea is that you excise the tumor, and these are tumors that Liz is developing the protocols over in Eduardo's lab. And so these are actual tumors. She sent me this picture, I think on a Saturday one day when she was in and got them. And then the T cells are able to be extracted, and then she's going to be transducing them with a gene that the Davila lab has worked on over the last decade and has demonstrated can make TILs into what we now call mighty TILs. So it's mighty 88. It's a component of the NF-kappa B pathway that allows the cells to survive and really be highly functional and highly expansive. And we think it's going to be, they're much more functional and much more potent in animal models and kind of in vitro assays. So right now, the GBF, we're really working up kind of the tech transfer process, being able to make everything GMP compliant, turning processes like this that are very complex from sort of a research grade into what can be done in the GMP suites is quite the project. So I just want to wrap up by saying a little bit about, I think, the amazing opportunities that GBF has, and this is why I was excited to come and join the team. I think thinking about, I was obviously working at cell therapy within the context of a startup and then in big pharma, and figuring out sort of how cell therapy can fit into the big pharma machine. The answer is not super smoothly. Part of it is that the GMP manufacturing for cell therapies is so different from small molecules or even biologics, and it's, you know, unique and has so many critical processes that require deep understanding and deep experience. I think it's fantastic that GBF has such direct access to the clinic. I think it gives opportunities like dosing patients with a fresh product, with having patients come onto study quickly, certainly access that big pharma doesn't have, except through collaborations where it's really throwing things over the fence and it's not a kind of collaborative effort. The direct access to academic collaboration, it's been so great working with Eduardo and getting to be involved at a very early preclinical stage. I think it was somewhat unique that I got some experience with that at pharma. I think it depends on what pharma company you're with and how much they want to collaborate with academic groups. But I think that academic to pharma collaboration is absolutely critical for these highly nuanced, highly innovative therapies to make their way successfully into patients. And then adaptability, just that GBF has the capacity to run. It was something that I was pushing for hard in board meetings and things at Gilead, where I would go in and say, they would say, okay, but what's version 5.0 of this? And I would say, I don't know, we need to run, 10 phase 1 clinical trials to iterate and get to the best curative product. And they would kind of grumble and laugh. But I think GBF is in a place where that could be possible to really innovate and adapt and run successfully run a bunch of phase 1s and really contribute to this field where really a lot needs to be learned clinically. So I think we're in a position to start to think about how we can address solid tumors using cell therapy. TIL manufacturing, to a large extent, is really just going off of what Steve Rosenberg developed, I don't know, starting 20, 25 years ago. All the major centers are still just using his protocols. So I think there's a lot of space for advancing manufacturing. We want to iterate on next generation TILs, like through Eduardo's project and through other next generation CARs like the 19x22. Manufacturing cell types beyond NK and T cell. We've talked to a bunch of exciting different potential collaborators about different cell types. I'm super excited about things like CAR macrophages. I think there's just a lot of opportunity. The immune system has so many cells in it and they all need to work together. And then incorporating some of these novel editing platforms beyond viral, I think is something that we can easily, not easily, but maybe more easily than other groups, put some effort behind and be real leaders in the field. Okay, I'm done. Thank you so much for this opportunity.